Okay, my name is Wade Holland. I'm a graduate student at Baylor College of Medicine. I'm in the cell and molecular biology program there, and I'm currently studying diabetes. I'm doing my PhD. Today I'm going to be talking to you guys about homeostasis, which is the regulation of the internal Mallu. So what is homeostasis? When we refer to homeostasis, what we're talking about is having constancy in a system. Okay? When we talked about earlier, we were talking about the normal resting state. If you think about the normal resting state as being how you want to be, when you have a disruption in your normal resting state, you will eventually come back to that state. All right? And that's what we call homeostasis, is the ability to adapt to change and return to a normal system. Physiologists will refer to the term homeostasis as regulating your internal environment. Okay? Because we don't have any say much in our external environment, but our bodies can control their internal environments. A homeostatic system is going to make adjustments to lessen the internal impact of major external events. All right? And an example that we know about now is exercise. When we exercise, our heart rate goes up, our breathing rate goes up, our blood pressure goes up. But as we stop exercising, we return to normal. We would not be in a very good state of being if our heart rate stayed the same at resting as we did when we were exercising. Plus, when we're exercising, our demands are greater, and so our body has to change to meet those demands. So we keep a constant supply of oxygen and such. So what do I mean by the internal Mallu? The internal Mallu is just a fancy term for meaning the extracellular fluids that surround your cells. It's the internal environment of an organism. These fluids provide a place for cells to deposit waste. It is a place where nutrients are absorbed from the extracellular fluids into the cells. And cells are the simplest units of life. They come together and they make up our homeostatic systems. Body systems are responsible for maintaining homeostasis. And as long as we have homeostasis, then we can have healthy cells. Another way of stating this is that maintaining homeostasis is dependent on the cells. All right? Each cell contributes to maintaining homeostasis. But at the same time, the cells are dependent on homeostasis. So it's kind of like a circle. The cell needs a constant internal environment. It can't deal with major fluctuations. Cells need to obtain nutrients and remove waste from their bodies, from inside them. And when we talk about cells and different systems, we have to realize that all cells have different functions. Okay? The cells of our cardiac system have different functions than the cells of our digestive systems. And they contribute differently to maintaining overall body homeostasis. So we know that homeostasis is this constancy in a system, but in actuality, it's a fairly relatively new term. Claude Bernard is a French physiologist from the mid-1800s, and he did a lot of work in describing this internal environment, getting the concept out that the internal environment of a body is going to stay relatively stable. And he recognized that certain organisms are able to maintain this constancy. Walter Cannon is a prominent American scientist. He coined the term homeostasis in 1926. He also started realizing that certain systems play very big roles. For instance, the central nervous system plays a huge role in maintaining homeostasis. So when we talk about maintaining an organism's internal regulation by homeostasis, what kind of different things are we talking about? Well, we have a constant body temperature. When you go to see a physician, he takes your temperature. He's making sure that that's staying in the normal range. Concentration of waste products. If waste products get built up in your blood system, you can have serious defects. You can have kidney failures. Gas exchange. We breathe in oxygen. We breathe out carbon dioxide. If those balances are altered, if we get too high of a carbon dioxide concentration in our blood, we can have severe problems. If we don't have enough oxygen, our brain can die. pH, energy requirements. Obviously, we need energy to do all of our basic tasks. Water and ion balance, volume and pressure, our blood pressure. If we have too many wastes in our blood pressure goes up. Homeostasis is trying to get that back to a normal range. There's two types of organisms that we need to consider then when we're talking about homeostasis. We need to talk about regulators and we need to talk about conformers. You and I are regulators. We have mechanisms in our body that are able to adapt to change. So if all of a sudden we go jump in a freezing cold pool or something, our bodies are going to adjust to that sudden change. Other types of organisms called conformers don't have those kind of problems, don't have a way to deal with those kind of issues. Basically, their internal environment is going to change with their external environment. So what they have to do, because they still can't manage major switches, major changes. So what they have to do is they have to live in pretty stable environments. So microorganisms that live in a certain concentration of salt water, that doesn't change too much, things like that. But we are considered regulators. And so for the rest of the talk, I'll pretty much be talking about regulators. And so then we're talking about homeostasis. Well, what makes up a homeostatic system? I've been saying all this stuff about body systems and homeostatic systems. What makes up a homeostatic system? Well, you have three necessary components of a homeostatic system. You have a receptor, which can also be considered like a sensor, a control center, which is going to process information from the receptor and determine what kind of action needs to be taken. And that action is then performed by the effector. In this diagram, you'll see that if we're thinking about our living environment, the room right now we have a thermometer that's reading what the temperature is, and it's constantly feeding the information to the thermostat. The thermostat will recognize at a certain point that, all right, it's getting a little too warm in here, I need to cut on the fan. So now the fan, or the effector, gets turned on, and eventually the temperature will cool. At that point, our thermometer says to the control center that we're back to our normal temperature, and the control center is going to shut down the fan. These kind of things are happening in our bodies at all times. So what's the mechanism of that system? It's actually got a very specific name, and that's negative feedback regulation. Because when the temperature in our house got back down to a normal system, got back down to a normal level, we don't want the fan to continue blowing, because then it's going to shoot way past what we intend it to be at. It's going to get too cold. So by the temperature being regulated, by the thermometer then telling the control center that the control center should shut off the fan, that's negative feedback. And we see this in our body all the time. Now, if we have negative feedback, do we have positive feedback? And we do. But positive feedback is not a part of homeostasis. The best example of positive feedback is live birth. When the uterine wall contracts, those contractions are continued through a process of positive feedback, meaning, okay, the contractions are going, let's keep going. And it keeps going until the baby is born. Okay? I study diabetes, so I know a little bit about glucose homeostasis. What diabetes is a disruption in glucose homeostasis. So I'm going to talk a little bit about that to give you guys some real-life biological examples of a homeostatic system. So think about two things. You can have basically one or two things here. We can have a high blood glucose system, or we can have a low blood glucose system. And we obviously want to be right in the middle, right? We want to have normal blood glucose. But think about a high blood glucose sugar content for right now. This can be toxic to the cell, so we can't allow this to happen. What happens is glucose transporters that are expressed on our beta cells get glucose from our blood and bring it into the beta cell. That's our receptor, is the glucose transporter. Our control center is the beta cell itself. And what the beta cell does is it then makes insulin. And insulin will be the effector. What insulin does is it's secreted from the beta cell, and it goes out to peripheral tissues like your muscle cells and your fat cells. And it tells those cells, hey, we have a lot of glucose in our blood right now. Let's take that in. And so we lower blood glucose by having our peripheral tissues take in this glucose. Our muscle cells are going to use it for energy. Our fat cells are going to store it away for a rainy day. On the other side of the ball, we have low blood glucose. All right? We obviously can't have this because if we have low blood glucose, we don't have enough energy. And if we don't have enough energy, then we can't go to school. So what happens there is we also have another glucose transporter. But this one is only on the cells, on the alpha cells of the pancreas. So glucose is sensed by, the low glucose is sensed by that alpha cell. And the alpha cell, in response to this low glucose, and the alpha cell is now a control center, is going to secrete the enzyme glucagon, or the hormone glucagon. What glucagon does is it goes to the liver, and it tells the liver that it needs to produce glucose. And the liver has mechanisms of doing this. And so what happens is we raise the blood glucose. But as you see, we have a system where we can lower blood glucose, and we have a system where we can raise blood glucose. And these systems are on at all times. And so actually what ends up happening is we have very little internal change in our blood glucose because these systems are constantly interacting.
So when we exercise, what happens?
Okay, when we exercise, what happens? That's a great question. First of all, we need to have energy, right? But there's only a certain amount of energy, and we can't, I don't know about you, but when I'm running, I can't sit there and eat constantly while I'm exercising to get that extra fuel. What ends up happening is our body maintains a normal glucose level, but we will actually go a little bit low, and what ends up happening is the body will switch to alternative forms of energy. We might turn to burning fats. Eventually, we'll turn to burning muscles. We will turn to alternative energy sources, but the energy source is maintained. So low glucose is not toxic to our cells. It's just not going to allow us to have enough energy. High glucose, on the other hand, is very toxic to us, and so we can't have that. So this is just another way of saying this. If we eat food, we end up having high blood glucose for a second. All right? Beta cells respond to this, secrete insulin. The excess glucose is taken in by the cells, and we return to lower blood glucose. Lower blood glucose could also be times in between meals. When it gets too low, the alpha cells are going to start secreting glucagon, and our liver is going to start making glucose for us. Yeah.
What is the raw material for the liver to produce glucose?
Excellent question. Glycogen, there's actually a couple of different processes that the liver can use. Gluconeogenesis or glycogenolysis. You can break down glycogen into glucose. There's other ways that the liver can actually manufacture glucose. But the main source is glycogen. And that's usually the storage form of glucose. When we're not using it, it's going to be converted into glycogen. And it's stored in the liver. It can also be stored in muscle cells. So we've talked about maintaining homeostatic systems. The problem is these systems can get disrupted. And when they get disrupted, we have to fix them. So how can these be disrupted? Injury. If we have a punctured lung, we're obviously not going to be able to obtain the amount of oxygen that we need, and that has to be corrected immediately because our brains cannot survive without a proper amount of oxygen. And if our brain goes, everything else is going to go. Illness is another way of disrupting homeostasis. If you go to the doctor, like I said before, and he takes your temperature, if you have the flu, you're going to have a fever. Now, a fever is a great way of a body responding to infection. But if we can't return to normal temperature, a high fever can destroy your nerve cells. It can eventually kill you. So we have to be able to control our body temperature. And illness is a way of disrupting our ability to do that. Disease, we'll talk about diabetes more. Disease is another way of disrupting homeostasis. We can think about the genetic components of diseases. So some people have a genetic predisposition to diabetes. So they have a genetic component that's going to lead to a disrupted homeostatic system. On the other side, lifestyles can help bring on disease. And thinking about diabetes, if you see somebody that's not exercising or not eating the right foods, we see that they have an increased chance of getting diabetes. That's very simplistic, but that's basically what it boils down to. Right now we're in the middle of a diabetic epidemic, and what we're seeing kids with diabetes. Now we've always seen kids with diabetes, but they've usually been considered type 1. Now we're seeing type 2, and what that is this is obese kids. And we're seeing kids that are not exercising nearly enough. We're seeing kids that don't eat right foods, junk food, sodas, those kind of things have become a staple in these kids' diet and lead to severe problems. Oh, and then death. Obviously, death would be the ultimate form of disrupting a homeostatic system. But the real reason I put that down is that these other things, if a homeostatic system is disrupted and not allowed to return to normal, death is a likely outcome. So let's talk about diabetes. Type 1 diabetes mellitus, also called DM. If we think about what we talked about earlier, high blood glucose detected by the beta cells release insulin, peripheral cells take in this extra glucose, and we return to normal. It's just a continual cycle here. This is how the body deals with it. In type 1 diabetes, what happens is we have an autoimmune reaction, meaning antibodies produced by our bodies recognize beta cells and signal their destruction. So the beta cells are no longer existent in a type 1 diabetic. So they have no insulin factory, which is what a beta cell really is. It's just an insulin factory. If they don't have that, then these peripheral tissues never get their signal, and our ability to regulate blood glucose is diminished. These steps do not happen in a type 1 diabetic. How do type 1 diabetics survive then? Well, prior to the 18th century, they typically didn't. But now we have ways, because thanks to our ability to produce insulin and give insulin shots, a diabetic can regulate themselves and live long, normal lives. Type 2 diabetics are a little bit different. So there's multiple ways of disrupting the same homeostatic system. Type 2 diabetics, what we see is that over time, the peripheral tissues, the fat and the muscle cells, they become resistant to insulin. Insulin is still being produced just fine, coming and talking to the peripheral tissues, and the peripheral tissues are just not listening. So they don't take in this extra glucose. So what happens? Over time, because the cells are becoming more and more resistant over time, what happens is that the beta cells, over time, start producing more and more and more insulin. So what happens if something is extremely busy for a long period of time? It gets tired, right? Eventually, the beta cells will fatigue. And so now their ability to produce insulin diminishes greatly. And what happens is, over the period of a lifetime of a type 2 diabetic, we go from normal blood glucose in their early lives to maybe a midlife, being pre-diabetic, and elderly having a much greater chance of getting to the stage where they're extremely insulin resistant and at the same time not producing much insulin at all. And so type 2 diabetics have a really hard time later in life in managing their glucose homeostasis.
What determines their resistance to insulin? Like, does it happen normally?
That is a great question, and you can't. Insulin resistance is still something that's not totally understood, but one thing that does lead to insulin sensitivity is exercise. So when we're seeing this type 2 diabetic epidemic that we're in right now, we're thinking that the lack of exercise is playing a huge role in these people's peripheral tissues, because exercise actually sensitizes you to insulin. But there's other things. The amount of fatty foods, fatty acids present in the body, that can decrease the amount of sensitivity to insulin, things like that. It depends. Now, this is kind of a timeline of where glucose homeostasis is. So, and the purpose of this slide is basically to show you that we are making enormous strides recently. The first description of diabetes came in 1552 BC. It's the first written one that we've been able to find. In the 1st century AD, it was described as the melting down of flesh and limbs into urine. Frequent urination is a symptom of diabetes. So we knew that even way back when, people that were medically interested in medicine understood something that was going on in these individuals. There was something that was askew. But it wasn't until 1776 that Matthew Dobson showed experiments that it was sugar in the blood and urine of diabetics. The way people used to be diagnosed as diabetic was actually the urine was actually tasted. And if there was a sweet taste, they knew that this was probably what was happening. 18th century, along comes Claude Bernard, the guy we talked about earlier, that really said, the internal environment of an organism is important in homeostasis. Claude Bernard showed studies of the function of the pancreas and the liver and how they affect homeostasis. In 1869, Paul Langerhans identifies the cells, the beta cells and the alpha cells, of the pancreas, but didn't really know what they did. And then they showed that if you pancreatize a dog, if you take the pancreas out of a dog, he's going to develop diabetes. So now they know this organ system is really important in maintaining our glucose. And then 1920s, let me just say that at the middle 18th century, that's when it became what's called or 19th century, that's called the experimental period in diabetes research. That's when they really started doing a lot of experiments trying to dissect out what these people understood to be happening but didn't know why. In 1921, insulin is discovered. By 1923, the people that discovered it had already won the Nobel Prize. It was already being used as an effective treatment for type 1 diabetics. Now, the way they were producing it was at first through cadavers. They were extracting the insulin from cadavers. Eventually, they moved into using pigs and using pig insulin to treat humans.
So they removed the blood of the cadavers and got the insulin out?
They could, yeah. In 1983, the biotech boom started hitting, and they were able to use bacteria to make insulin. And what this did was it made us, we don't have to raise pigs, so there's no much reduced feed and space costs. We didn't have to dissect cadavers, which is just probably a horrendous job anyways. What we did was we grew big vats of bacteria and extracted out a very pure insulin. So it became cheap for everybody. And so type 1 diabetics had a nice, cheap way of treating their disease. And then lastly, 2001, the human genome sequence was announced as being finished. There's still a lot of work to be done in annotating the sequence, but this sequence is now what's really driving research in diabetes.
What were the problems when there was when they used pigs?
What were the problems?
I mean, besides the fact that you had to grow the pig, were there a lot of rejection as far as, or is insulin close enough to pig?
Insulin is a small molecule, so it's probably not going to be considered able to be rejected because, you might still have, and when we have an autoimmune reaction to the beta cell, it's recognizing the beta cell, not insulin. Now, if you have antibodies to insulin, exogenous sources of insulin are not going to help you anyways. So currently, where is diabetic research? Clinical trials. These things are going on all over the nation, all over the world. Physiological traits of patients, response to therapeutics. There's all new drugs that are coming out all the time that they are getting evaluated. We've heard the things about Celebrex and things like that. It's not always a good response, so we have to be extremely careful. So these clinical trials are really important for the health of the patient. But to get to that point, we also need to know a lot about the disease, how it comes about. And so there's genetic approaches. We can look at the function of particular genes that we think are playing a role. We could look at the insulin gene itself, how it's expressed from DNA to RNA to protein. We can do family-based studies. That's an important way of determining how diabetes is passed from one generation to the next. We can do what's called genome-wide scans. At that point, genome-wide scans is a very high-tech way of looking at a person's entire genome and looking for abnormalities. They can compare a normal person and a diabetic person and do things like that. We have animal models now. We can make mice. We can make other animals do what we want them to do genetically. We can build transgenic animals. And these are great sources of information for this kind of study. And then microarray analysis. This is something I put on here because microarrays are new technology, and basically what it is, it's also like a genome-wide scan. It's a really high throughput way of looking at a lot of genetic material in a very small space. They are extremely informative and are yielding a lot of new evidence on what's happening inside of a diabetic cells. So future, this is kind of short because there's a lot more work than just 2 bullet points in diabetes. But continual utilization of the human genome sequence data. This is what I'm doing now. We need to be sequencing people from different ethnicities. We need to make sure we have good coverage because what might affect an African American individual might not affect a Caucasian or an Asian. So we have to have this kind of, and we also see discrepancies. In the United States, African Americans and Hispanics are the 2 most likely to be affected by diabetes. So there's something going on there that yields important data for all of us. And then beta cell transplantations have been in the news a lot lately. This is where beta cells from a healthy individual have been transplanted into a type 1 diabetic. So you're thinking these will be destroyed. What ended up happening are these severe type 1 diabetics who were taking insulin multiple times per day to regulate their glucose, all of a sudden were medicine-free. Now, as it turns out, eventually these cells do die off and they're back on meds. But a first step has been taken, and so it's a really exciting new technology. So just in review, homeostasis refers to the ability to maintain a constant internal environment. If there's one message I want you to take home, that's it. That's what a homeostatic system does. It regulates and maintains a constant internal environment because our internal environment can't stand big changes that are caused by big changes in our external environment. Illness, disease, death, injuries can all lead to disruptions in homeostasis. We've talked a lot today about diabetes because that's what I know a little bit about. And that's a disruption in glucose homeostasis. And it's only one example of many of disruptions in homeostasis. And then lastly, as a little optimistic view, science is actively pursuing a broader and more in-depth understanding of homeostatic systems. And they are able, in our search to be able to treat patients suffering from disease or illness, injury, etc. Any questions?
Yes, I have a couple of questions. In the previous slide, you showed that one of the therapies is aimed to transplanting beta cells.
Mm-hmm.
Is there any treatment aimed to counter all the immune cells that are attacking the beta cells?
So I'm not an expert on that part. I do not know. There are ways of suppressing immune systems. We do it all the time when we have other kinds of transplantations. For instance, if you were to get a kidney transplant, you would be on immune suppressive drugs. So they very well could be on a cocktail of those kind of drugs. But the problem is these people are naturally going to express that. So they might have to take these immunosuppressive drugs for a while. What we know is that in the long term, currently beta cell transplantation in the long term is not a viable and permanent fix. But that first step showed a lot of promise. And so they're going to continue with this, and I'm sure that they're going to get to a point where this is a routine treatment for type 1 diabetics.
Yes. And this is not directly on diabetes, but why do diabetic people don't heal the wounds, and why can they become blind? What is the link?
Okay. Wound healing, blindness, renal failure, non-traumatic limb amputations, these are all complications of diabetes. What it is it's a consequence of the high glucose toxicity. If the concentration in our blood of glucose gets too high, that is toxic. It will destroy your nerves. It will destroy your capillaries. So when you're saying, oh, I'm not healing a wound, well, if your platelets can't get to that wound because your circulatory system has been damaged, your wound healing is going to be slower. That same glucose toxicity can destroy the nerves, the optic nerves and things, and stuff like that. That's what's happening there. That's a complication of diabetes. Capillaries, very small blood vessels. Basically, the end of the line as far as blood, the blood system or the circulatory system is concerned. Go big arteries, arterioles, all the way down to really tiny, single cell wide capillaries. Any other questions?
How does glucose transporter can control both alpha and beta cell?
Excellent question. It's not that it's controlling it. Basically what it is, it's a doorway to those cells. So the reason I say it's a receptor is, it's just like if I'm throwing you a baseball and the baseball is glucose, your glove is the glucose transporter. It's just going to catch that glucose. It doesn't really do anything else. It just lets it into the cell. And then the beta cell or the alpha cell could say, all right, I'm not seeing nearly enough, or I'm seeing way too much of this. So I got to do something.
Has there been an increase in type 1 diabetes in kids? And what are the primary causes of that? Why
Depending on what population you look at, there have been some increases in some populations of type 1 diabetics. For the most part, though, the big increase that we're seeing is in type 2. Now, what causes type 1 diabetes is an autoimmune reaction. The other thing that we're seeing is that, there could be a possibility that the reason we're seeing more type 2 diabetics is that our ability to diagnose them at an earlier age has increased. So that could be one thing. We're paying more attention to that now, although we are seeing a true shift from old to young. But in type 1, there's environmental components to that disease as well. And so we don't know what all those are. So if there is an increase, I don't know what's causing it.
It's more genetic, though.
Type 1 diabetics typically is thought of as being more genetic. However, it's still a very complex disease. So there's multiple gene interactions. There's multiple environmental components that go into type 1, just like maybe not the environmental aspects possibly could have or probably are having a much bigger role in type 2 diabetics. But there's a genetic and environmental component to both syndromes.
Now, I'm going to switch a little to the evolutionary aspect of that. Why would we see this shift in our bodies that go for, because you see more and more now type 2 diabetes. So what is the shift to select for genetic aspects that would make us more prone to diabetes?
That is an excellent question, and what you're getting at is what's called the thrifty hypothesis. So we live in a time now of abundance. If you and I are hungry, we can go to fast food, we can skip over to the grocery store and buy something that's probably not too healthy for us if we don't want to. But centuries ago, this was not the case. Food was scarce. So when you had food, you ate it, right? Our bodies developed mechanisms of dealing with high intake, but not over a long period of time. Now we're having high intake over a long period of time. Plus, and so what we have is we have genetic aspects there. Our ability to deal with the presence of abundant food. The other thing is manual labor, our jobs back then, how we couldn't jump in a car and go somewhere. We would have to walk, run, ride a horse, do something active to get from point A to point B. You can get by now with fairly little active processes. And that is not helping as far as diabetes is concerned. I mean, you've got to exercise. You've got to eat right. But that's a great question. I mean, you can take diabetes and look at it as an evolutionary system too. Homeostasis, you can look at that as an evolutionary system. Because these things, our demands have changed over time.
How does metabolism fall into all of that?
That's a great question. Metabolism differs between people. Some people are going to have an extremely high metabolism. Some people are going to have extremely low metabolism. Those people have to alter lifestyle. They're probably not going to be able to alter their metabolic rates. So they're going to have to alter something else if their homeostasis is getting out of whack. If their glucose homeostasis is getting out of whack, they're going to have to cut down on the amount of food they eat or select different types of foods. But a person's ability to deal with what brings in, their ability to metabolize things, is a huge component of diabetes. A huge component of all homeostatic systems.
And it changes with age, correct? So your metabolism tends to slow down after a certain age and just continue to slow down.
Right. Parts wear out, if you want to think of it like that. So that's why, and that's also why we say early on in life, having young kids getting type 2 diabetes when we have normally only seen that in older people. Imagine what the complication rates are going to be for these kids. There's a possibility that this generation will not live as long as their parents, which is a horrendously scary thought. But it's a definite possibility. Okay. Thank you very much.